Hello everyone. As many of you know, one of Noxopharm's major milestones was being part of a team that published a breakthrough paper in the journal Nature Immunology, a paper that for the first time revealed the detailed science underlying our technology. The paper was led by Professor Michael Gantier at Hudson Institute of Medical Research and a high-caliber international team of researchers. It's a very important paper for our company, so let's take a look at the key points. The most important finding in the paper is our discovery of a critical natural mechanism that helps prevent our immune system from attacking our own body, which was previously poorly understood. To explain in more detail, Toll-like receptors 7 and 8 are key sensors or alarms in our immune system that detect the presence of viruses by sensing their RNA. When this happens, TLR7 and 8 trigger an immune response to help destroy the virus. However, when this natural system misfires, these receptors can sometimes mistake our own RNA, which is very similar to viral RNA, as a threat. This then leads to the body attacking itself, as seen in autoimmune diseases like lupus. So the question is, why isn't our body constantly getting triggered by our own RNA? The paper shows that specific tiny RNA fragments within our own body, containing a special chemical tag called 2'-O-methyl, act as natural regulators and are constantly being produced, essentially turning down or blocking the activity of these TLR7 and TLR8 immune sensors, acting as natural brakes, if you like. These naturally occurring ultra-short RNA pieces can be very small relative to natural RNA and RNA-based therapeutics, and directly interact and bind to TLR7 and TLR8. This binding puts the receptors in an inactive state, preventing unnecessary inflammation against our own RNA and helping our cells avoid autoimmunity. In fact, it has been observed that patients with rare mutations in TLR7 and TLR8 that make them resistant to this protective effect develop severe autoimmunity as children due to these receptors being constantly in an active or switched on state. This discovery, therefore, solves the long-standing mystery of how our immune system maintains balance with 2'-O-methyl modified RNA fragments serving as built-in brakes to prevent disease. This revolutionary new understanding also opens new possibilities for novel therapies by mimicking these natural RNA fragments to treat autoimmune diseases through calming overactive TLR7 and 8 signaling. In summary, these tiny 2'-O-methyl RNA fragments are natural guardians that limit harmful autoimmunity by inhibiting TLR7 and TLR8, as well as other immune sensors that we will talk more about in future. That is the discovery underlying our Sofra platform, and from there we're building a library of potent assets that are attracting interest from around the world. Thank you.